Good morning, everyone, and welcome to the second webinar in the iBridge Plus Spotlight series being hosted by NIBMG. So this webinar series is being organized by the iBridge Plus Institutes to inform public about the key scientific outcomes from our research. For today, our speaker is Dr. Moulinath Acharya. Dr. Acharya is an associate professor at our institute. His research interest is in complex traits associated with vision and neurodegeneration, and in understanding the genetic heterogeneity in oligogenic diseases with muscle or neuronal defects. Today, he is going to talk about his recently published work in PLOS Genetics on functional investigation on one of the diseases that they had found through the CWAS studies in his lab in primary angle closure glaucoma. So, Dr. Acharya, may I please invite you to deliver your talk.
Thank you, Alpana, for this kind introduction. Good morning to everybody. And let's start without much delay. Let me share for my presentation first. I hope this is visible to everybody. So, as we know, glaucoma is basically a heterogeneous group of optic neuropathies with complex genetic basis, and this is the leading cause of irreversible blindness worldwide. So what happens in glaucoma is basically a progressive degeneration of retinal ganglion cells and their axons, resulting in the loss of visual field. And the global age standardized prevalence is 3.5%, which is among 40 years and older. And there are common subtypes like angle closure glaucoma, open angle glaucoma, congenital glaucoma, and so on. So we are interested in primary angle closure glaucoma, which is have highest prevalence in Africa. And so POAG is one type of glaucoma, and angle closure glaucoma is another major subtype, which has the highest prevalence. POAG has the highest prevalence in Africa, and PSEG has a prevalence of 1.1% in Asia. So this is like a bilateral blindness is more prevalent among the angle closure glaucoma patients than open angle glaucoma. So what exactly happens here? Let me show a pointer so that you would understand it better. So here, this is the ciliary, this is basically the sagittal section of the eye where you have the lens. Where you have the lens, and this from there, this aqueous humor actually originates from the ciliary body, which actually comes through this anterior chamber and exits through this trabecular meshwork, which is placed between iris and cornea. So what is the requirement of this aqueous humor? The first requirement, since eye is an anterior segment of the eye is an avascular organ, so it provides nourishment to the adjacent organs like the cornea, iris, lens, etc. And also it maintains the anterior chamber structure. So there is no problem as such with the production of the aqueous humor, but if the drainage is blocked like here, then it actually causes a raise in the intraocular pressure. Intraocular pressure is IOP, which is regulated by a balance between secretion of aqueous humor and the drainage of the aqueous humor, okay, through this Schlem's canal. So obviously, if the drainage is not proper, the IOP will raise, and which in turn will put a pressure on the posterior side of the eye and thereby causing a loss of retinal ganglion cells and thinning of the retinal nerve fiber layer. Here in this picture, what we are looking at the fundus of an eye, where you can see this is a normal eye where this is the disc and this is the cup. So it is like a hollow pipe, okay? So the cup is the hollow part and the disc is the rim. So as you can understand that if the pipe becomes more hollow, that essentially means that the rim is becoming thin. So that happens in glaucoma, and if you look at the ratio, if we consider the ratio of cup and disc, so this is called the CDR or cup to disc ratio. As the cup to disc ratio tends to be a 1, you have seen that glaucomatous damage has happened, and as a result, there's a vision loss. So what we have done is genome-wide association study in primary angle closure glaucoma. So before moving into that, I would like to spend a few minutes on the study design. If you consider this Indian population, and I have already talked about the iridocorneal angle between the iris and cornea. So this angle, when it becomes narrower than 15 degrees, that essentially means that you have a angle closure starting point. Okay, it's not an angle closure. We call it an occludable angle or narrow angle. If you can look at the data, this is a published report where it says that 30% of the normal population is actually having these occludable angle or narrow angle. So this is not angle closure though. Okay. So what we thought that for CWAS to be successful, you need to reduce the heterogeneity. So to make it more homogeneous, what we thought that we are looking into the genes responsible for glaucomatous neurodegeneration. So we are not interested in the angle related genes. So why not taking all individuals, they are having a kind of narrow angle so that we could minimize that the genetic enrichment for the angle related parameters and maximize the genetic enrichment for glaucomatous neurodegeneration. With that intention, we did that. But then there is one caveat. The caveat is, what if these narrow angle individuals will not progress to glaucoma? What is the guarantee in that? So nobody can say that. So for that, since angle closure glaucoma is a late onset disease, what we thought is, if we choose these patients that are having glaucomatous neurodegeneration a little bit early onset, like 40 years or older, 40 years or younger, and our controls, if we select 60 years or more, then that would basically, because if they are having occludable angle and they have crossed 60 years of age, there is a minimum risk that they would develop glaucoma. So with that intention, we have segregated our patient and control cohort. Now, if you look at our patient and control cohort, the intraocular pressure is significantly higher in the PSCG subjects than in PSCS. PSCS is the primary angle closure suspects, which they never develop glaucoma because they already passed this age. Okay. Now, and also for cup to disc ratio, you can see that the cup to disc ratio is much higher, which is statistically significant in the glaucomatous group. Now, the question comes that how would we get these individuals, because these individuals will not come to the clinic. They are normal individuals, right? So, yeah, absolutely, they would not visit the clinic for their eye problem, but at the age of 60 years, there is another problem called cataract, and that happens. So people often visit the clinic, eye ophthalmology clinic, for cataract testing. So we actually thought that we would look into those population, and if we see that their angle related parameters are 15 degrees or below, because according to this data, 30% of the Indian population is having these narrow angles. So we would get sufficient number of controls. So that's how we designed our study. So this is the extreme phenotype GWAS design. So this extreme phenotype, meaning this is not a phenotype. Age, we considered age as a phenotype, which is basically a proxy of this phenotype. Okay. And then we did association testing. That's the way to do it in GWAS. I will come in a little bit details on this part later. And then we have identified variants associated with PSCG and related quantitative traits. The quantitative traits we are not discussing today, and we will more focus on the risk factors for glaucomatous neurodegeneration. So before we going into that, let's first look into what exactly a genomoid association study is. So in genomoid association study, what we do is data collection and then genotyping. And then we need to look at that this genotyping, we are doing it in a case control design. So we need to see that our cases and controls are having the same genetic ancestry or not. So for that, we need to do the quality control with so that basically says the genetic relatedness between the populations. And then we do imputation, which is basically it's not possible to do this is a genomoid genotyping array. So it's not possible to put all the SNPs in the genotyping array. So there are databases available like 1,000 genome data. Now we have Genome India data. There you have already informations, a lot more informations are there for other SNPs. So that can be imputed into your study. So that's how it can be designed. So this is a statistically heavy process, but it can be done. And then after that, we do is association testing. This meta-analysis is a additional step, but it is not absolutely necessary to do a meta-analysis, because if you are doing it for the first time, you might not have a control data from the other cohorts or other populations. But the most important part is replication. Replication is whatever you have found from the discovery GWAS, you need to replicate those in a separate cohort, at least some of these SNPs, so that they would stand the same association that has been happening in the discovery phase. And then comes the post-GWAS analysis. I will spend a little bit more time on the post-GWAS analysis. Post-GWAS analysis is basically to look into that what are the associated loci, and then looking at what are the likely causal variants and what are the epigenomic effects of these variants, and what are the target genes in that locus. So everything is actually post-GWAS analysis, and everything is very important. And finally, the long-range interaction and the pathway analysis. So what we have done is a haplotype-based GWAS. A haplotype is a particular pattern. Those who don't know about a haplotype is a particular pattern of sequential SNPs which are found on a single chromosome, and it has a block-wise structure separated by hotspots. Within each block, the recombination is rare due to tight linkage, and only very few haplotypes really occur. Okay, so this is basically a haplotype. And what we have done is basically to make it more robust, we have done it a haplotype-based GWAS, where you see that we have taken 3 SNPs, 4 SNPs, 5, 6, 7, 8, and onwards. Beyond 8, we have not found any association. So until 8 SNPs, we have done this for. So beyond that, because this now, if you put 9 or 10, it becomes very big, and the haplotype is kind of lost. Okay. This is the workflow. I'm not going into the details of the workflow because I have already explained in the previous slide. So what we have done is for these 3, 4, 5, 6, 7, and 8, we have done a logistic regression on these haplotype blocks, and you can see that these this is a Manhattan plot, and you can see that this is the genomoid suggestive threshold, and there are these SNPs are always on top of these genomoid threshold for all these haplotype blocks. Looking closely, we found there are almost more than 35 or 39 SNPs are there, and they are basically associated different, like say for example, this is the first SNP that is being found in 3 haplotype structure, 4 haplotype, 5, 6, all the haplotype structure we have found it. Likewise, this is the table that you can see it here. So those SNPs are basically, these are the SNPs that are found in all haplotypes. And then the next set is found in 4, 5, 6, 7, 8. The next found in 5, 6, 7, 8. And then so on. Okay. So altogether, we have 13 SNPs that has been found in the genic region called CNTNAP5 or CANTNAP5. So the often in these association studies, there is a sentinel SNP, which is basically the SNP that shows the maximum association, which is this one here, 780010. If we look closely in the 780010, this regional association plot shows that it is the high statistical strength compared to other regions. So when we looked at the clinical correlation, I have already spoken about the cup to disc ratio, and you can see the cup to disc ratio in a more detailed manner. So the cup to disc ratio less than 0.5 is healthy neuron, and if it is more than 0.5, then this is kind of a diseased neuron, we would say. Uh, and retinal nerve fiber thinning is happening. So if you look closely, this is the data that I have already shown that between PSEG and PSES. But what about our sentinel SNP? The sentinel SNP actually do show association, significant association with cup to disc ratio as well. Now, there are, as I earlier said, that there are 13 CANTNAF5 SNPs, and there are other SNP also. Altogether, there are these many numbers of SNPs are there in the haplotype blocks. So this 13 SNP signature, when we looked at the LD plot, there is a clear two blocks are happening here. Okay, so you can see these two block structures. So these are the two haplotype blocks expanding these 13 SNPs, and this actually remains there when we looked at their open chromatin structure using the screen tool. Okay, this is a tool in the is that uh, UCSC genome browser. You can find it. This is the ENCODE database. So this essentially says that there are peaks. Those peaks are for CTCF binding, DNase hypersensitivity, H3K27 acetylation, and H3K4 trimethylation. So these are all suggestive of open chromatin structure. So, and you can see the first block, which is in blue, these SNPs are not having any great peaks here, but the red block having these number of SNPs, they are actually showing better open chromatin structure, which is also corroborated in the next picture, where we have used a deep learning based algorithm framework for predicting the chromatin effects of sequence alterations with single nucleotide sensitivity. And this is the multiple testing, the stringent multiple testing correction, Bonferroni. And if you look at the SNPs, this is our sentinel SNP, the RA780010. And this structure, the haplotype block structure that was there in the LD plot, that has been actually, that shows the same structure in the DeepSeq database also. And there are these red blocks, those SNPs, there are SNPs that are really having effects on this, okay? So, so we have taken these 2553628 for our further analysis for functionalization. So we did a conditional analysis using this 2553628, and this SNP, we found that this is really significantly associated. And they have a CTCF binding site and a transcription factor binding site as well in this region, TCF4. And what we have done next is allele specific activity of this SNP. And we have found that indeed forward these, they have shown a different allele specific activity for different alleles. The luciferase activity is shown. So this is more like an enhancer activity because this SNP is present in an enhancer region. When we looked at the long range interaction of these whole region actually in the CANTNAF5, we found that using this huge in database, and what we have done is we have not found any retinal cell or retinal data. So what we have looked into is the neural progenitor cells and dorsolateral prefrontal cortex area, which both are the brain area. And then you can see that there is a topologically associated domain in the CANTNAF5 region here in blue. Okay. And in the dorsolateral prefrontal cortex, this region is also there. And in addition to that, there is a frequency regulating element that's actually sitting inside these loci. So where exactly these SNPs are. And when we looked at the CANTAP5 gene expression from the GTEx data, so these are all almost concentrated to brain region. And GTEx usually do not have the eye data. So we have used the eye integration data for looking at CANTAP5 expression in the different eye tissues. And you can see that in healthy retina, there's huge expression, normalized expression in CANTAP5 expression, which is also corroborated in the human protein atlas data, where you can see in the retina, actually more CANTAP5 is there than the other brain regions. Okay. So at this point, we thought that since this gene or genic region, the total gene is showing so much association and I mean in all aspects like uh, the long range interaction and the expression data, all everything is pointing towards this. So why not we try to knock down this gene and see the effect of this gene in zebrafish. So what we have done is using a antisense oligo. This is called morpholino. So it has a morpholin ring. That's why it is called as morpholino. So essentially, this is an antisense oligo technology where you design the antisense oligo against these genes, and then this is a translation blocker morpholino. So what it's having is basically instead of the phosphodiester, these antisense oligo will have a phosphoramidate bond, and which actually forms a stable structure with RNA and blocks this initiation complex to proceed further, and thereby blocking the translation. Okay. So we have used, since most of the genome in zebrafish is natural duplication happens. So we have two orthologs of human CANTNAF5 in zebrafish, CANTNAF5A and CANTNAF5B. And like all morpholino studies, we have used a 5 base pair mismatch. So essentially this, what happens in this mismatch is basically 5 different areas are mismatched so that it is used as a control. So we have different mismatch control for CANTNAF5A and CANTNAF5B. So here we are looking at the phosphorodamidate, this is the phosphodiester bond. And this is basically what happens in morpholino, which blocks this initiation complex to form. And in a single cell stage, if you inject that, this basically blocks the expression of CANTNAF5 protein. So as I said, we have used CANTAF5 mismatch control and the translation blocking morpholino into single cell zebrafish embryos. And if we look at these eye structure, the CANTAF5 morphan show a fairly significant reduced eyes, which is corroborated in multiple experiments. We have done more than four, but we are showing here 4 sets, where you can see looking at how many we have injected, how many total death, and how many were having phenotype, and etc. Also, we have done these analysis where we have compared between morpholino and mismatch control. The injected and total death is not significant, whereas obviously the ocular phenotype, there are other phenotypes also. They are pretty significant, and the normal phenotype is much more significantly higher in mismatch control, which it should be. Now, when we look deep in the structure of the retina upon this CANTNAF5 knockdown in zebrafish, we saw a disrupted retinal cytoarchitecture. So these are the different retinal layers you can see. This is the ganglion cell layer, and this is the inner plexiform layer, inner nuclear layer. So these layers are intact in the mismatch control, whereas these layers are all disrupted in the CANTNAF5 morphin, which was not obvious from the way it was when we injected, just looking at from the outside. What we did next is basically we have designed antibody for these zebrafish CANTNAF 5. This is a customized antibody that we have designed, and with this, we have actually immunostained these sections, the sagittal sections of the embryo. And if you look at both this mismatch control and morphin, the morphin barely show any expression of CANTNAF5, which is also statistically significant here. Next, we have looked at a very interesting phenomena. This is before going into that, let me first explain what is alpha tubulin or alpha tubulin acetylation. So alpha tubulin acetylation actually restricts axon overbranching by dampening microtubule plus end dynamics in neurons. This is the paper published in 2018. So this acetylation acts as a brake for axon overbranching and overgrowth by dampening these microtubule dynamics. So this is totally related with the neurodegeneration, the tubulin acetylation. Now, when we use these as a we did immunostaining with alpha tubulin, acetylated tubulin, we saw that in the mismatch control, barely you can see the expression, and whereas in the morphin, the expression is much, much higher, which is also corroborated in this diagram. We have done multiple times this analysis. Next, we have looked into the nasal granules. So nasal granules are basically, they are the chromatolysis agents. So if you have more chromatolysis, the nasal granule will be less there. So that essentially shows the neurodegeneration. If you look at mismatch control, the nasal stains or neurotracer staining is perfectly fine, whereas in the morphin you can barely see it. So this again proves that there is neurodegeneration that is going on in these morphins. Next, we have looked into the apoptosis because in glaucoma there is neurodegeneration and there is a huge apoptosis that has also been observed in glaucomatous condition. And that actually leads to this neurodegeneration. So what we have observed is pretty interesting. So we have done these cleft caspase. Cleft caspase is a marker for apoptosis, and we have used whole mount embryos to actually look into these cleft caspase expression. If you see these data, that these mismatch controls and the morphin, they look very similar, but the apoptosis is much higher all throughout the body in the morphins. And if you look closely, they are much higher in the eye and the brain region, which is also significant from this data. Now, when we looked at altogether, we looked at the DAPI. DAPI stands for the nucleus. And if you look at the CANTNAF5 expression, the acetylated tubulin expression in the mismatch control is much lesser. In comparison, the morphin shows similar DAPI expression. The CANTNAF5 expression is almost nil, and then huge increased expression of the acetylated tubulin structure. And finally, this is the merged picture. And also, we have done Western blot in zebrafish live from the zebrafish lysets, and you can see that there is a huge difference in the acetylated tubulin expression in morphin. What we have done next is looking at the locomotory behavior of the zebrafish, because if they are not, their vision is problematic, that should be reflected in their locomotory behavior. What we have done is basically this is not a video, but we have taken the video. Unfortunately, I could not place this here, but what we have done essentially is a 6-well plate where these top panels are all mismatch controls and the bottom panels are all CANTNAF5 morphins, and we have looked into their locomotory behavior, okay, in the dark and light cycles. So how did we do that? So first you need to habituate them for 60 minutes or 1 hour and then you put them in the light and dark cycle for 10 minutes. And this cycle, so what we have done is basically four light and four dark cycles. And in there, you can see that each of these light cycles, look closely in the light cycles. As we move forward, the light cycles, you can see the locomotory behavior, more locomotory behavior for the mismatch control, whereas the morphand remains same, which is actually depicted in this picture, where in the light phase, there is a huge difference between the morphands and the control, mismatch control, whereas in the dark phase, this is not no significance. They are behaving completely normal. Now, we are actually relating these behavior with their vision problem. I know this is not the best way to look at that, but in future, we will use another method called optokinetic response to look into this behavior more closely. In summary, what we have done is basically a haplotype-based GWAS, which identifies CANTAP5 association. And we have done conditional GWAS to find out, so these SNPs basically prioritize the SNPs, and which shows the histone modification and transcription regulation and so on. And then we moved into zebrafish to knock down the CANTAP5 gene, and we observed the disturbed axonal transport, disintegrating microtubule structure, which is kind of evident from the acidotubulin staining, and which ultimately leads to the degeneration and healthy difference between the healthy and degenerative retina. And we have also looked at their vision-guided locomotory behavior. This work is recently published in PLOS Genetics, and I will just briefly, I have a few points to mention here. And another study, so same GWAS, actually, we have used that GWAS data to look into the copy number variation structure, where we have found that, and we have done qPCR to validate that too. We have found a new gene called NLGN1 or neuroligin 1, that those are amplified in PACG patients and not present in the PACS. So this is very interesting data. Why this is interesting, I'm telling you in a while. So now the question comes, the obvious question is how to use this knowledge in future. So what we are planning using these CANTAP5 and NLGN1. So we will be dissecting the role of these CANTAP5 and NLGN1 interactions in the synaptic maturation and retinal neurodegeneration. So you can see this structure. This CANTAP5 is a type of neurexin, which actually acts through this calcium signaling pathway, and NLGN1 or neuroligin 1 is basically guiding through these postsynaptic membrane NMDA receptor. So what we are asking, the questions are, first question is, how does CANTAP5 regulate tubulin acetylation in retinal neurons? Does CANTAP5 physically interact with NLGN1? If yes, what is the impact of altered CANTAP5 NLGN1 interaction on synaptic plasticity and retinal neurodegeneration? And the final is whether modulating this tubulin acetylation, we can rescue or exacerbate this retinal neurodegeneration. So in summary, this is the future goal where we are looking at the neurotransmitter release of these using neurexin, CANTAP5 as a neurexin and NLGN1 is the neuroligin. And we are actually planning to create NLGN1 and CANTAP5 transgenic zebrafish to look into the acetyltubulin and the other experiments that we have done for our PLOS Genetics paper. And finally, this vision-guided locomotory behavior, not only we will use these the scientific system that we have right now, but we'll devise something that we can also measure the optokinetic response. This is mostly this study was funded by the intramodal funding from NIBMG, and also partially by one of my previously funded grant from Department of Biotechnology. And this is Dr. Sudipto Chakraborty is already awarded the PhD degree. So Sudipto has done mostly this work, and Sudipto was funded from ICMR. So this image is basically showing the whole structure, the coronal structure of the zebrafish embryo, where you can see two eyes and the brain region. And as I can, so this green is CANTNAP 5 and red is acetylated tubulin. So you can see that in brain region there are acetylated tubulin expression. But if you look at CANTNAP 5, that is mostly concentrated in the retinal ganglion layer in both these eyes, retinal ganglion layer of this region. And we have these small group here. So this is the neurodegeneration research group. With that, thank you very much for your attention.
Thank you, Dr. Moulinath, for this wonderful talk and very nicely breaking down things for our audience so that they can understand the nitty-gritties of the study that you have done. So for our online audience, you can always put your questions in the chat box, and they will be addressed now. If you have any questions, I had one which Dr. Moulinath answered at the end, that how is he going to take the study forward and what are his future plans with it. But for the online audience, if you can just put your questions in the chat box, and we will, Dr. Moulinath will be able to answer them. Or the other option is, if you think of these questions later, you can mail it to him or to me, and we will get back to you with the answers. Or you can reach out to us on our NIBMG social media handles, and we will address questions. Okay, so so there is one question from Mr. Praveer Varman. What did you get from CNV?
What did I get from?
CNV.
Sorry, I could not hear it.
What did you get from CNV?
CMD.
CNV.
CNV. Yeah. So CNV is a copy number variation, as I have earlier pointed out. So so what is the question? Like, is it how we can did the CNV analysis?
I think how was it used? Yeah. How did they?
Yeah, because CNV, I deliberately didn't show the data because I need to bring neuroligin 1 in the context. So just I showed the excerpts of the CNV data. So essentially what we have done is basically we have done a genomoid genotyping to get this the association study, right? So this genomoid genotyping data is essentially a DNA microarray, right? Where you have the different SNPs there in the array, and then you do a genomoid genotyping. So those genotypes you will see because if you look at the green, that's homozygous for the major allele, and then green red for homozygous for the minor allele, and if you have yellow, that means they are heterozygous. So that's how this is done. But now, if you look closely, like the previous RNA microarray in the DNA microarray also, these structures are basically these intensities of these red, green, yellow, you can actually measure it. Okay. So this copy number variation, actually this analysis is based on this intensity measurement. So that is converted to the logger and the beta value, and that you can actually use for copy number variation analysis as well. The same genotyping data. It has been done quite, I mean, in all over world, it has been done quite nicely. So it is nothing new that we have done. So this is something that actually naturally comes from these. When you do genome-wide genotyping, you can actually use this to look into the copy number variation. So that's how we have done it.
So, and then how did that analysis of the copy number variation help you with your with the correlations that you have done?
Yeah, so essentially these are two different studies. Okay, so essentially CANTNAP5 has nothing to do with the copy number variation analysis. We have not found CANTNAP5 from our copy number variation analysis. So rather we have found it from the conventional GWAS, the haplotype based GWAS, and also for the CDR quantitative trait analysis, the CANTNAP5 gene. And three different modes we have found. But when we looked into the data, the CNV data, this NLGN1 amplification we have seen in PSCG patients. Those were not there in the PSCS controls. So this is a separate data which we have done further on the qPCR of this total region of NLGN1, and we have found that this is actually true. So the amplification is really happening. So that data I have already shown in the slide. The bottom box plot was basically that qPCR data. So this we have communicated. Let's see. This is under review right now. So let's see how it. I mean, what kind of comments we receive for this manuscript.
Sure.
Anything else? I hope not.
Not. We don't have any more questions in the chat box yet. So,
okay.
Thank you, Dr. Acharya, for your talk, and thank you for everyone who has joined online. Please do look out for these. We'll continue to host these webinars as and when we have a publication or any other research output, which should be shared with the public. So do look out for these webinars. So thank you, everyone, and
thank you very much.
See you next time.
Okay.